Advisory Committee and affiliate faculty member of the EEB to have been asked to introduce Dr. McLaren today for her seminar. Jenny joined us in 2014 and she has basically catapulted herself, her lab, our students, and our university into the stratosphere. Really some outstanding research focusing on global change biology. And she'll be talking to you a lot about that. Her work primarily focuses on nutrient cycling of nitrogen and phosphorus and the like, which are fundamental constituents of all of the cycling patterns that contribute to changes on a global scale. And what's really wonderful about Jenny's work, as you'll see, is that it takes her and her students both to far-flung places like the increasingly green snow slopes of Alaska to the increasingly creosote-invaded deserts of New Mexico. And she'll be showcasing both her local and her distal work on this. Jenny has set up an amazing research infrastructure of talented graduate students. She has 4 graduate students right now and 2 that she has seen to completion. She has authored 14 peer-reviewed publications in outstanding and in many cases exceptional journals, such as the Proceedings of the National Academy of Sciences and submissions to journals like Nature Communications. She's also generated, for her research enterprise, $750,000 in funding, and she has also worked on many international collaborations and I'm learning from her on how to create an international network, and I have so a long way to go. I'm really in admiration of the international relations across many different laboratories for very difficult longitudinal experiments that Dr. McLaren has been conducting to really represent as an ambassador of UTEP in the best possible way. So her talk today is titled The Importance of Plant Identity in a Changing Climate. Jenny?
All right, thanks Arshad. And I want to start out too just by thanking my graduate students and my undergrads. They produced most of the data that I'm going to show you today, and so I want to make sure I acknowledge them kind of right up front. So it's hard to escape the news these days that the Earth's temperature is rising. We know that the global temperatures are rising about 0.2 degrees Celsius per decade, but we also know that this isn't happening in even patterns around the world. The Arctic is warming a lot faster than other regions are, and this is one of the reasons why the iconic polar bear and other types of Arctic mammals really are the poster children for climate change. But climate change is only part of a broader phenomena that we're seeing, where we're having really large-scale, global-scale changes to the environment caused by humans. And these encompass something that we call global change. Global change encompasses things like increasing plant resources that you might see through agriculture fertilization or emissions from the air because of oil and gas exploration. Humans modify the environment through the way that we manage the land, such as putting grazers on land or clear cuts of forests. And then finally, humans also directly change the species that live in ecosystems by moving them between different continents. And this has resulted in things like species invasions. Now, global change has resulted in a lot of really large-scale consequences that we both experience and hear about in the media. Things like increasing fires in California over the last few years, but also we're seeing fires in the Arctic tundra now where we didn't see them before. They're having effects on things like species diversity and abundance. And if you saw in the news just last week, there were big reports coming out on studies showing that we've lost 1/3 of the birds in North America over just the last 10 years. And then finally, global change is causing species to move as well. And so as the environment warms, species are typically moving further north or higher up in elevation as they track the temperatures that they're most adapted to. And this is another one of the reasons why the polar bear has become an icon of global change, because some animals actually have nowhere further north to go. So the consequence of a lot of these kind of landscape scale changes that we're seeing is that it's creating novel communities, or when we go to an ecosystem, the types of species that live there now are not the same species that lived there in the past. So global change drivers lead plant communities to change with the different types of species or different abundance of species. Two examples here that I'll talk about a little bit today. You can see one, these are historical photographs showing the same landscape in Alaskan tundra. Especially if you focus in the foreground here, you can see that the number of woody plants or shrubs is really increasing in Arctic tundra. If we look at an example here from Sonoran Desert, photo from the 1800s, the Sonoran Desert and locally the Chihuahuan Desert were grasslands during this time, but over time have shifted to either cactus-dominated or creosote-dominated deserts. Now, when these plant communities change, the way the ecosystems work changes as well, because different types of plants have different influences on ecosystem properties or functions, such as productivity or how much carbon these ecosystems fix. They may change the types of animals that live in these ecosystems. Things like carbon storage, both above and below ground, or nutrient cycling. And so when these functions change, one of the reasons as researchers and as humans we're interested in this is ecosystem functions can also be directly linked to what we call ecosystem services, which are basically functions that humans are interested in. So things like productivity and nutrient cycling are linked to the services of food production or fertilization needs in agriculture. If we're not storing carbon below ground, it may be emitted to the atmosphere, which can affect global temperatures. And then if the types of animals that live in communities change, this also affects things like disease transmission, because when those animals move, the diseases that they carry or the vectors that they carry move with them. And so looking at these shifts is one of the things, or is the main thing that my lab does. The motivating questions that drive the research of my research group is firstly, when we're looking at this shift, when global change drivers happen, what happens to the plant community? How does plant community structure change? And then when plant community structure changes, how does that change the way the ecosystem works? Or how does it affect ecosystem functioning? So today I'm going to tell you about 4 different areas of research that my lab's been working on, focusing first on some collaborative research networks that I've been part of using work that I've conducted in northern Canada. I'll tell you about some work I've done on shrub encroachment, or that my research group does in shrub encroachment in northern Alaska, northern Canada, and in eastern New Mexico. I'm doing a lot of work looking at indirect changes to plant communities through things like increasing resources. And so I've got experiments there running again in northern Alaska, northern Canada, and also in Carlsbad, New Mexico. And then the final topic I'll talk about is looking at kind of an even more indirect change to plant community structure, which happens when you change the herbivore communities, which then change plant communities, which then changes the way that ecosystems work. I'll finish off by talking about some of the future directions that I'm planning on taking these research areas, and then briefly summarize some of my accomplishments at UTEP. So to begin with, over the last few years especially, I've become increasingly involved in these collaborative research networks. So one of the inherent difficulties with manipulative ecology or manipulative experiments like I do is typically you have to go to one place at one time to run the experiment. And it can be difficult to say whether the results that I find in northern Canada in these few years are going to be applicable to other areas of the world at other times. And so there's a few different ways that ecologists have used to get around these problems, with one of them being meta-analyses. So a meta-analysis is when you can combine data from many different studies kind of after the fact, after they're already published, and see if we're getting similar results across different ecosystems. One of the difficulties with a meta-analysis is that the experiment's already done, and so people have used different experimental methods or different ways of measuring response variables, and it makes it more difficult to combine these studies afterwards. So I will tell you about a meta-analysis I've been part of lately called CORRE, but then I'm also going to talk about some globally distributed experiments, and there's been a call for globally distributed experiments instead of meta-analyses, because these are something that are kind of done before the fact. So different researchers will go out, design an experiment, and you do the exact same experiment in many different locations around the world at the same time. And by coordinating our methods, we have a lot more power in being able to increase the generality of our findings. So I'll talk about one of these types of globally distributed experiments I've been involved with in a warming experiment. But to start out with, I've been part of this research group called CORRE. It stands for Community Responses to Resource Experiments, and it's a network of researchers who manipulate global change factors. So to be part of this research group, you had to have an experiment in a grassland that was manipulating at least one global change factor. You'd been measuring plant community responses for at least 3 years. So this resulted in, we've got 105 different experiments and about 70 different researchers combining our results. There's experiments on all grasslands in the world except for Antarctica. And you can see that the age of the experiment ranges between about 3 and 30 years. So the experiment that I contributed to this is an experiment I've been running in northern Canada. It's been running not only by me, but for 30 years now. So this is one of the longest running experiments in this meta-analysis and one of the longest running experiments in the world. I used this data, or this experiment was established using funding from NSERC in Canada, but over the past 5 years I've been maintaining and sampling this experiment using primarily funding from UTEP. Now, the data from this experiment combined with all 104 other experiments, some of the major things that we've found so far, the first one really surprised us actually, is that when we're manipulating global change factors, so things like nutrients and temperature, we're not actually losing species from this experiment like we predicted, but what's happening is we're changing the identity of the species that are found in these plots. The second thing was that was encouraging is these grasslands were resilient to global change, but to a point. Experiments that were less than 10 years old, we only saw changes in plant community structure in about a third of the experiments, and the other 2/3 showed no change. Whereas if the experiment was over 10 years old, over 70% of the experiments showed a significant change in community structure. So the other thing that this really highlights for us is the importance of running these experiments for the long term, because global change isn't just happening for a period of 3 or 4 years. So these results just came out last month in PNAS. Now the second type of collaborative research I've been involved with is one of these globally distributed experiments. You can see this truly is a globally distributed experiment. We have 10 different locations around the world, and I'm the lead PI for the Northern Canadian location here. This is a group called WARM. It stands for Warming and Removal in Mountains. And like I said, we all install a experiment that exactly the same type of experimental manipulations in multiple locations. So at all 10 of these locations, we have warming manipulations set up using these greenhouses, and these warming manipulations happen at both high elevation sites and low elevation sites at all of the 10 locations. We have a plan to warm these plots for 5 years. Next year is going to be the 5th year of the experiment. We've been measuring plant and soil responses as we go, but at the end of the experiment here, we have planned a synchronized sampling, which we're expecting to result in a number of really high impact synthesis publications. We did just this summer submit the first publication from the first 2 years of data here. It's currently in revision for Nature Communications. So to finish up the collaborative networks here, since arriving at UTEP, I've published 4 papers with collaborative groups. Number 3 here is actually with another collaborative group that I didn't tell you about today. A number of undergraduate and graduate students have helped me maintain these experiments over the last 5 years. And then really large group of collaborators here on these experiments. But in addition to these collaborative networks that I work with, I also run a number of independent research streams in my lab, with one of them focusing on the effects of a plant community change that's already happening, or shrub encroachment. Now, if you've heard me talk before, you may have heard me talk about shrub encroachment in the Arctic before, but shrub encroachment or woody plant increases is actually happening in many different ecosystems around the world. And so I'm going to tell you about 2 different areas that I work in. One is Arctic and Alpine tundra, and then the other is locally in the Chihuahuan Desert. Now for the tundra work, shrubification or increasing shrubs in the Arctic has been a topic that people have started looking at a lot recently because it's being observed circumpolar. So if we look here, you can see areas in green. This is a view of the world from the top. These areas in green have seen an increase in vegetation productivity over the last few decades. So there's more biomass being produced, and a lot of this increase in biomass is thought to be because the plant composition is shifting to one dominated by these larger woody shrubs. On the ground measurements have shown us, again, many locations circumpolar have seen observations of increasing shrubs over the last few decades. Now you can see that we think that this increase in shrubs is actually going to have a lot of different effects on these Arctic ecosystems. There's global scale feedbacks here, regional scale feedbacks, but what my lab really looks at are these local scale feedbacks, or how they might change things like nutrient cycling and microbial activity in the soil. In particular, one of the things that really interests me is how plant communities influence ecosystem properties. I've phrased it before as in the afterlife, or really once they've lost their leaves. How did the plant litter influence ecosystem properties? And in particular, how does the litter influence the way that decomposition happens? And so when plant communities change, two aspects of their litter change. The first is the quality of the litter might change. So in the Arctic, we're getting a replacement of these types of plants, so evergreen shrubs and graminoids, by deciduous shrubs. Deciduous shrubs have different carbon to nitrogen ratios, different amounts of lignin, different properties that may cause them to decompose differently. But in addition to shifts in the quality of the litter, we're also seeing shifts in the quantity or the amount of litter being produced. Again, in this ecosystem, many of these species actually retain their leaves for multiple years. And so I started thinking about this question and when I first started working in Arctic shrubland, and I just saw this thick litter accumulation underneath the shrubs that you don't see in other areas. So some undergrads of mine have documented this in Alpine tundra, showing about 4 times more litter in areas with dense shrubs than in open tundra. And so to answer the question of whether litter quality is going to influence the way that carbon and nitrogen is cycling back into this ecosystem, we want to look and see how quickly this litter is decomposing. And so in a study I had come out in Ecology a few years ago, we looked at how these different species decompose. Right along here, we have these deciduous shrubs. You can see they're losing litter a lot faster than the other 3 species, which are dominant species that are being replaced by shrub encroachment. But I think answering the questions about litter quality are actually a lot more difficult, because as shrubs are increasing, litter is increasing, but they're inherently happening at the same time. And so it's really hard to tease apart what are the effects of the quantity of the litter versus what are the effects of the shrubs. My 2 graduate students can tell you how hard it is to tease this apart. So this is a project that was started by Alejandro Benumea and then is being completed by Daniela Aguirre. And here what they've done is they've manipulated independently the presence of shrubs and then the presence of this litter. So half the plots have shrubs and half the plots they've clipped all the shrubs away. And then within these they've actually gone out every fall, collected litter from the ground and redistributed it between these plots so we can independently measure litter density. This experiment ran for 4 years in northern Canada. So Daniela is getting ready to graduate this semester, hopefully, and so she sent me some of her data that I could show today, with this first one looking at how soil temperature is changing in these plots, which we think will directly affect the way that litter decomposes. So one of the things that really surprised me when I saw this data was that the amount of litter on the ground doesn't actually change soil temperature, despite the fact that it looked like it was a thick insulative barrier. But what really does change soil temperature is the presence of the shrubs themselves. So in the winter months here, you can see that when the shrubs are present, you actually have a warmer soil temperature, so microbial processes can be happening faster. This is something that's been shown before. It's called the snow shrub hypothesis, that when you have that physical structure of shrubs in an Arctic landscape, they collect the snow, and those deeper snow layers result in insulation for the soil below. But what hasn't been shown before is these summer differences here, where in the summer months you're actually getting cooler soils. This has been hypothesized before because the shrubs are actually shading the soil and lowering soil temperature for the microbes there. So when we looked at how quickly the litter are decomposing in these different environments, the first thing we saw is probably directly because of the soil temperature. Up here in green, you can see areas that do have shrubs, and they are decomposing at a slower rate than areas without shrubs. So those cooler temperatures are likely slowing down those processes. In red, we have areas without shrubs which are decomposing faster. But we're also finding effects of the amount of litter there on how quickly things decompose. And so depending on whether there's shrubs or not shrubs present, increasing litter either increases or decreases decomposition rate. So you can see there's a lot of interactions here happening in the analysis, and Daniela and I spent about an hour yesterday actually trying to tease some of these things apart. So just bringing back up this diagram about how shrubs may be changing ecosystems and walk through some of these hypothesized arrows here. The first one is we did find, as a few other people have, that increasing snow depth leads to more winter insulation and higher soil temperatures. But we also showed that shading in the summer is likely what's leading to these lower soil temperatures in the summer. You can see that shrub litter was hypothesized a few years back to be more recalcitrant than the litter that it's replacing, but we found that that's not true. Actually, the litter decomposes more quickly. It's got a higher quality than the litter that it's replacing. But then we also found that you get an increase in litter quantity here, but this is having questionable effects on how the litter is decomposing. So most of my work on shrub encroachment has been in Arctic ecosystems, but lately we've started adding in some of this research in the drylands, because the drylands, in a really similar way, are experiencing this shift from a grass-dominated ecosystem to a shrub-dominated ecosystem. Where in the 1800s, locally, we were dominated by perennial grasslands, but through a variety of different factors, a lot of it may be due to overgrazing. When Europeans first settled this area, there's been a shift towards these creosote-dominated deserts. So this shift towards a desert ecosystem has led to a project that's being run by the USDA and the Bureau of Land Management called Restore New Mexico, where the goal is to go in and do these large-scale shrub removals using an aerial herbicide and try and restore these grasses. So I'm not going to show you data from this today, but Kathleen Schaefer has started doing her PhD project working with the USDA, but rather than looking at whether the grasses are recovering, she wants to see how these large-scale shrub removals are actually affecting the ability of the desert to retain soil carbon. And so follow up in a couple of years with Katie for how this works out. So in the shrub encroachment section, since arriving at UTEP, I've published 3 papers, with some being authored by UTEP and non-UTEP undergrads and graduate students. 3 different graduate students working on this project, lots of different undergrads. You'll see this as a common theme in all of my experiments. And then I've had a number of collaborators on these projects, both not locally on my Arctic work, and then locally with Craig Tweedy and Brandon Bestelmeyer on the desert work. Okay, so when I'm looking at shrub encroachment, I'm looking at plant communities which have already shifted. But one of the things that I'm also really interested in is how these plant communities shift when we are experiencing global change. And in this way, one of the things that I'm looking at most is when you change plant resources, how does that change the way that these ecosystems are structured? So we know a lot of different types of plant resources are changing with global change. Things like the amount of nutrients in the soil, water, precipitation, even the amount of carbon dioxide in the air. I think of these, manipulating plant nutrients is one of the most common types of manipulations you'll see ecologists do. One, because it's way easier to fertilize than it is to change the amount of rainfall a landscape is getting, but also because we think that most plant communities are actually limited by the types and amounts of nutrients that they have. Now, changing nutrients has led to some known community shifts in different types of ecosystems, where Arctic tundra, typically as you increase nutrients, you get an increase in the shrubs present in this ecosystem. In temperate ecosystems that have more rainfall than we do here, usually you'll see an increase in grasses as you increase nutrients. In drylands, it's a lot less known because dryland ecosystems are typically thought to be limited by water instead of nutrients. There's many fewer experiments manipulating nutrients in these ecosystems. Now, in the Arctic, nutrient manipulations, at least in Arctic Alaska, have typically focused around manipulations of nitrogen. So in the 1980s and 90s, Terry Chapin and Gus Shaver did a series of fertilization experiments, some of the first fertilization experiments in this area. And they showed that the Arctic tundra shows really strong responses to nitrogen, but there were some hints of co-limitation by other nutrients. But because the responses to nitrogen were so strong, subsequently, there's been a major focus on nitrogen for both our experimental treatments, but also what we measure, kind of to the exclusion of other nutrients. And this is especially true in terrestrial ecosystems in the Arctic. I think one of the things that really highlights this is this experiment here that was set up by the Arctic LTR 27 years ago, where it's a factorial experiment. You've manipulated nitrogen and phosphorus independently as well as together. The majority of the papers that have come out from this 27-year-old experiment have focused only on these 2 plots, just asking what happens when you fertilize, and the majority of those responses are attributed to the effects of nitrogen, without actually separating out what was the effect of nitrogen and what was the effect of phosphorus. I think what really highlighted the neglect of phosphorus in this ecosystem for me was these phosphorus addition plots have been manipulated for 27 years. as far as I can tell, not only have they never been published, but no one had ever sampled them before. And so a collaborator and I, Kate Buckridge from the University of Edinburgh, right when I started at UTEP, we decided this was a great use of my startup funding, was we got out there to sample these different plots. What we found is published in Ecosphere this year. I'm not going to go through, we found a lot of results here, but what I really want to highlight for you is phosphorus is important. That's my main conclusion from this section. These areas highlighted in blue, this indicates of the vegetation responses, vegetation groups that responded to phosphorus independently. Things in green means that if there was a response to nitrogen, it depended on whether there was phosphorus added or not. So phosphorus is important in how the vegetation responds, but you do see a lot of nitrogen responses as we expected. Now we looked below ground as well, so we have here different pools of nitrogen or different pools of nutrients that we can extract from the soil. We also looked at things like the microbial biomass, or using fumigations we can detect how many microbes are in the soil. And I also do a lot of work doing enzyme activity assays in my lab to look at the types of enzymes that these microbes are producing and how they're accessing nutrients in the soil. So the first thing I want to highlight is there's a whole lot of blue here. A lot of the below ground responses were actually responding to phosphorus rather than nitrogen. But what I really want to highlight is the response of the microbial biomass. So in plant studies, if you increase nutrients and the plants grow more, you say they were limited by that nutrient. It's the same thing for microbes. When we add phosphorus, you see a lot more microbial biomass in the soil, and you don't see that when you add nitrogen. So what this highlights for us is that the microbial biomass is actually being limited by phosphorus rather than nitrogen. And it's just been assumed before that everything must be limited by the same nutrient. And so our main conclusions from this paper were phosphorus-specific responses and interactions are actually a lot more common than we expected. And most importantly, that nutrient limitation above and below ground may be different from each other. We have a second paper in review right now at Ecology Letters showing that the response of the microbial community and its ability to maintain its carbon, nitrogen, and phosphorus ratios actually may depend on the response of the plant community to fertilization as well. So most of the nutrient addition work that I've done has been focused on Arctic ecosystems. And in the Arctic, nutrient addition is really important because we think as Arctic soils are warming, they're going to start decomposing faster and increase their release of nutrients. But in more urbanized areas, or even in areas a lot further south, we're also seeing a lot of increase in nitrogen being put into these ecosystems. And some of this is because of oil and gas production and industrialization, which releases nutrients into the air, which then fall onto the ecosystems around them, which may be limited by those nutrients. You can see that oil and gas production in the US has actually doubled the amount of nitrogen emissions being released in just the last decade. And if you've gone to Carlsbad Caverns National Park recently, you've seen that there's a lot of oil and gas exploration happening right next to the park. That oil and gas exploration is going to be releasing nitrogen in the air, which may be landing on the sensitive desert vegetation right next door. So I had a project funded to look at the effects of this nitrogen deposition on both vegetation structure and also biogeochemical cycling in the soils, funded by the National Park Service. This is an experiment being run by Jennifer Olguin, who's a master's student in my lab. She's conducting an experiment at 3 different grasslands in Carlsbad National Park. She's about halfway through the experiment right now. She's actually leaving as soon as I'm done talking to drive out to Carlsbad and do the monsoon sampling for this year. And what we're really looking at in this experiment, again, is how these nitrogen additions are affecting the way the ecosystem works. So in the way she's adding nitrogen, she's actually doing it in a relatively realistic way. And so we're looking at control, which are receiving background nitrogen additions. And the highest level we're putting in is doubling the nitrogen emissions that we think are coming from the background, which again is what the US has experienced over just the past decade. We're right at the beginning of the experiment, but one of the first things we need to show is that the organisms in this ecosystem actually will take up the nitrogen that we put into the ecosystem. It's not just being limited entirely by water. So when we go up in the winter, and you can see all the different plots that we've added here, in the winter, there's not very much growing at that period of time, and so when we add nitrogen in these plots, you just see that nitrogen accumulating in the soil. But what's really encouraging for us is in the monsoon season, when you do get a lot more growth, you can see that those nitrogen levels are being brought back down, so the plants and the microbes in the system are very likely taking up that nitrogen, so they do have the ability to use more nitrogen than they're actually getting right now. So this has been the most active area of my research since arriving at UTEP. I've had 6 papers come out on nitrogen, or I guess resource addition, since arriving here, again with publications by undergraduate and graduate students as well. I've had 2 different graduate students work on these projects, again a number of collaborators and undergraduates too, and then Amanda Colts and I are actually talking this afternoon about the next experiment we have planned on these areas. May affect Arctic carbon cycling. Now, carbon is really important in the Arctic. There's a lot of us in this department that work in the Arctic, so you've probably heard us talking about how important Arctic carbon is, and this is because there's massive stores of carbon in the soil in the Arctic, and the ability of that soil to retain the carbon is going to be really important in controlling how much carbon is being released to the atmosphere. Now, a number of researchers have developed Earth system models in order to be able to predict how that carbon is going to be released to the atmosphere. And these models do incorporate the activities of organisms already. We really know that microbes are incredibly important in controlling how carbon is cycled, and also the plant community. And so those have been incorporated into most major ESM models. But despite evidence that these larger order organisms or basically animals can impact carbon cycling, very few of these models actually incorporate the activities of those herbivores. This has led to calls from a lot of researchers that it's time to animate the carbon cycle. It's time to incorporate the activity of animals into these models. So we do know in the Arctic that the presence of animals can control carbon, nitrogen, phosphorus cycling. A number of our colleagues have set up experiments where you put up fences and keep out these large animals, and we've shown that by trampling vegetation and eating vegetation, they're decreasing the amount of carbon this ecosystem takes up. But in the Arctic, these large animals, they're important, but they're only present for part of the year. Most of them are migratory. So when they're there, they may be there in large numbers, but they'll move on. Or if they're not migratory, they're typically there in fairly low numbers. Rodents, on the other hand, or microtine rodents, are ubiquitous in Arctic ecosystems. They can occur at very large numbers. So these Arctic rodents typically undergo population cycles where every few years you'll see really high densities of these animals. But they're also active year-round. So they're active through the winter under the snow. And we think that their foraging and other types of activities are going to have influences on the carbon, nitrogen, and phosphorus cycling in this ecosystem. So we proposed these hypotheses to the National Science Foundation, and we have a large collaborative research project that's been funded for 5 years to explore these questions. Austin Roy is a PhD candidate in my lab who's working on these questions, and we have set up a series of field experiments where we are manipulating animal density in the Arctic. This is an incredibly logistically challenging undertaking that we've been working on these past few years. We have 3 different locations that we're working at. So we have sites at Utqiagvik on the north coast, at Toolik on the north slope, and just outside of Nome on the west coast. And at each of these locations, we've set up 3 sites where we manipulate animal density. So we have treatments. We've basically built these large fences that are buried into the ground containing bits of tundra where we can change animal density over time. So we've set up 2 edges of the extreme. One is this press experiment where we are putting animals into these fences at high density over the entire 5 years of the experiment. We have a pulse treatment where this is more of a natural level of vole pressure. We're working with lemmings and voles depending on which ecosystem we're working in here. But the pulse means a high density of animals for 1 year, and then they're removed and we let the tundra recover. And so this mimics this natural population cycle. The other end of the extreme is exclosure, where we have no animals present for the entire experiment. And then we also have controls which have just background levels of animals. We'll be monitoring these plots for 5 years and measuring things like vegetation and plant litter, gas exchange, and different soil biogeochemical and microbial properties. And the ultimate goal of our experiment is to build a model. So we're working with a model called MEL. It's the Multiple Ecosystem Limitation Model, and it's a model that's already been designed to look at how plant, soil nutrients, and soil organic matter cycle nutrients and carbon through the Arctic. We have proposed a model we are calling AniMel that actually puts animals right into the center of MEL. So we're showing how the activity of rodents may be changing carbon and nutrient cycling in the Arctic. So last summer was the first summer that we had animals in the fences, everything was up and running, so we're still really early on in this experiment. So I'm not going to show you data from those plots yet, but what I am going to show you is we have evidence that small animals do impact carbon and nitrogen cycling. So this is some data that Austin, my PhD student, collected last year. We have 4 different activities that these animals do. So the rodents create runways. They typically use the same path over and over again to move through the tundra, so they're trampling vegetation in these areas. They'll create hay piles where they've chopped up vegetation. They use this as a winter nest and a food source over the winter. They create burrows. They use latrines, so they defecate in the same spot over and over, creating these piles of poop on the tundra. And what we've shown is if we compare things like the amount of nutrients that we can extract from the soil, the amount of microbes in the soil, or how those microbes are accessing nutrients through their enzyme activity, that we do see differences in the soil right underneath these different activity types. So looking just specifically at hay piles to begin with, you can see that underneath these hay piles here, we have higher amounts of nutrients. This is resulting in higher levels of microbial biomass, and those microbes are actually producing more enzymes in order to break down complex organic material to get the nutrients that they need. So for this project, it's a brand new project. We just submitted, or Austin just submitted, the first paper from his PhD thesis this summer. So I've got 1 graduate student working this project, a lot of different undergrads, not only undergrads that I take up to Barrow every summer, but we have a lot of lab work in these experiments as well. So we've got a team of undergrads working all winter to process the coolers of soil that we fly back with every year. So it's also a really large collaborative project. So these are my collaborators, the other PIs on the project. There's 6 of us total at 5 different universities. So just to summarize, I know I gave you a lot of information over the last 35 minutes. And so some of the take-home messages here, if I could get you to take home one major message from every group of experiments I told you about, for the first one in our ecological synthesis, I think it's really highlighting the importance not only of bringing together these large groups of researchers, but also of conducting our experiments over longer periods of time. Looking at increasing shrubs, we've shown that it's really important to consider how you might get feedbacks between changes in the environment and changes in the types of litter being produced. And these lessons we learned in the Arctic will likely be applicable to the work we're doing in the desert. When we increase resources, I think the most exciting thing that we found is that the above and below ground communities may be responding to entirely different sets of resources. And then finally, looking at the herbivore communities, even just really early evidence, we're showing that the effects of herbivores can cascade from animals to plants and then to soil. And so I have a few different future directions that I'm laying out here for where I'm taking these research areas over the next few years. So in terms of my collaborative networks, at Worm Network, we have our 5th year major sampling happening next summer. So we're really expecting a number of synthesis papers written over the next few years. Looking at shrub encroachment, I recently participated in a sampling of a 35-year-old experiment. I was in charge of the root component of this sampling and really participatory in the soils as well. We've just nearly completed our first manuscript, which should be submitted to Nature in the next month or 2, and then we have a second manuscript we're hoping to get out in the spring. Now, one of the things that I was really excited to find in the Arctic was this disconnect between the above and below ground communities. And so working with Amanda Colts, who's a specialist in below ground food webs, we're planning on submitting a proposal to NSF where we can use these long-term fertilization experiments in Canada that I have to really try and see if the entire below ground community, rather than just the microbes, is responding differently to the fertilization treatments than the above ground community. You've also seen that I've become incredibly interested in phosphorus over the last few years in most of the ecosystems that I look at. So one of the proposals we're looking at writing for the Arctic is to see how warming might affect phosphorus in the soil. Again, we know a lot about how nitrogen may change when we warm soil, but a lot less about what might happen to phosphorus. And so I've been working on this project with Kate Buckridge. It was a project I had Alison Nauman start, who did a master's certificate in my lab. And we're just completing some of the soil analysis to compare warmed and non-warmed soils and planning on using this both for a manuscript and then preliminary data to really explore phosphorus cycling in Arctic ecosystems. And then finally, phosphorus isn't only important in the Arctic, it very well may be important in the desert as well. Our REU project, we had some undergrads last summer, showed that desert microbes may also be phosphorus limited. And so Anthony Darzat-Nardi here in the department and I are putting together a proposal to submit to NSF looking at the importance of phosphorus in desert ecosystems. Okay, so I'm going to spend just the last few minutes here talking about some of the accomplishments I've had since arriving at UTEP. So since I arrived at UTEP, I've published 14 papers, and this is papers including both papers lead authored by undergraduate and graduate students in a variety of different journals. I also have 5 more articles that are either right now submitted or have been rejected within invites to resubmit very recently. So there's still a lot happening. And the other thing I want to point out here is that I'm really on an upward trajectory in my publication record. A number of the experiments I've showed you, you can see they take 5 years before we can start getting the data to start publishing those papers. And so the experiments that I started when I first arrived at UTEP are really starting to come out right now. And so many of these papers actually came out in just the last 2 years. In terms of my grants, I've brought over $750,000 to UTEP since I arrived, with a large portion of this being this Arctic herbivore experiment that we're running. So this is a collaborative grant. It's a $2.7 million collaborative grant, with over $600K of that came to UTEP. But I've also had funding from the National Park Service, the British Ecological Society, and then a variety of internally funded sources as well. We don't often highlight our failures, but I do want to highlight as well that I've also submitted 18 other unfunded grant proposals. A number of these include grant proposals that I've written with faculty at UTEP, and a number of them we're still working on and continuing to submit. In terms of my teaching, I have regularly taught 2 different undergraduate courses at UTEP, so both ecology and plant ecology. I'm teaching both of them this semester. And I think one of the things that really highlights not only my success in teaching, but the success of EEB as a group is our enrollment is really rising. So in ecology, I taught it my second semester here, and I had 19 students registered in the course, and I have 80 this semester. For plant ecology, I had 11 the first time I taught it, and now I've got 40 in the class. I've taught graduate course advances in ecological theory a few times since arriving as well. In terms of my mentoring, I've got 1 PhD student and 1 PhD candidate currently, and I've graduated an MS student, but have another MS student hopefully graduating this semester, and then Jennifer is about a year in. And I've also supported the research of a lot of different undergraduate research students. So I've had 54 undergraduate students in my lab since arriving. They've been funded through a variety of different sources. 20 of them have presented poster presentations either at CORRE or at international meetings. And then I've had 2 peer-reviewed publications with an undergraduate senior author and about 4 or 5 more in prep. Finally, just to finish off with my service here, I would say my main service contribution to the EEB group has been as a member of the advisory committee, but I sit on a number of other committees. I'm currently sitting on my 4th faculty search committee since I've arrived at UTEP, just showing how quickly our department is growing. And I've just finished up a term as faculty senator. But finally, I have also put a lot of service into the professional community. And I think that this is not only something that I really enjoy, but this is something that highlights UTEP on an international and a national level. And so I've done over 50 journal article reviews for 21 different journals in just the last 5 years, and I was recently asked to sit on the editorial board for Biogeosciences. Done over 40 ad hoc grant reviews for national funding organizations from the United States, Canada, Britain, and Israel. Sat on 9 federal grant review panels, and also served an elected term as the secretary for the Biogeosciences section. So with that, I'm just going to put up some acknowledgements. I really want to acknowledge not only my officially assigned mentors, but a number of informal mentoring that I've gotten from other faculty in the department. I've had a huge number of collaborators. You've seen a lot of them throughout this talk, and as well as some that I didn't talk about our research today. I can't emphasize my students enough that really without this, well, without them producing all this research in the lab, we wouldn't have gotten so many amazing results since I arrived here. And then with our far-flung fieldwork, I get a lot of logistical support and technical support on these projects. So I'm going to highlight funding and also just some support and patience at home as I go through these processes here. So with that, I'm going to ask for questions.
Questions from the audience?
Anthony.
I was intrigued by some of Austin's results. I was surprised that the latrines didn't have higher nutrients. Why do you guys think that is as compared to like the hay piles? I just would have predicted there would be a lot there.
I would have predicted there would have been a lot there too. I think in terms of at least available nutrients, that this is the nutrients that are available for plant uptake right away. And those nutrients are often really temporally variable. It's like you and I have a publication that shows in the Arctic how they vary a lot through time. But I think one reason might be, so I used to do herbivore work during my master's with geese, and we looked at how quickly the nutrients are lost from feces over time. This is one thing that Austin's looking at for the voles. But for geese, much of the nitrogen in their poop was gone in 20 minutes. So it may just be that under the latrines, the nitrogen's actually moving through really quickly, but it's something we're exploring further. We've got a lot of decomposition bags full of poop out there. Yeah.
So you mentioned that phosphorus is very important for the ecosystem, right?
Mm-hmm.
Particularly you were talking from the context of microorganisms.
Yep.
So you said that also that you were planning to submit a grant or follow through that project. So I'm not that surprised that there are changes because phosphorus is very important for the production of ATP.
Mm-hmm.
You have ATP, you have energy organisms, you increase the biomass. So what is your idea in terms of developing this project?
I think there's two things we need to consider. And one is, we're not surprised that microbes use phosphorus because phosphorus is really important in DNA and ATP and a lot of the different processes. Nitrogen is also really important in terms of building proteins and things like that. And so it's the relative importance of them that we're looking at. And I think the first thing that we really wanted to highlight for people is often when we fertilize the plots and you see a big response above ground, we just assume the entire ecosystem is responding in the same way. So if the plants do this, then the microbes must be as well. And so we're showing that that may not be true. And so I think one of the things that we're planning on pushing forward with here is just to look at, temporally, when is phosphorus really important? Is it going to change over these long-term experiments? We want to know to do it not in exclusion of the plants. I think one of the other things that's done a lot in soil microbial research is we look at these limitation experiments in a mesocosm. So some of the preliminary data I've showed is soil in jars that we incubate with different levels of nutrients. But the paper that we've got in submission to Ecology Letters right now is really trying to highlight the fact that the microbes are there when we add nutrients, taking up nutrients, but the plants are there at the same time and they're competing for those nutrients. And so it may be that the phosphorus is limiting these microbes because the plants are taking up the phosphorus better than the microbes are, and there's just not enough there for them. So I think separating out the role of plants versus the roles of phosphorus is really important. We've got a lot of different ideas where we're taking this, but in the Arctic specifically, we're really interested now in what's going to happen when you warm these soils to phosphorus cycling and how that might affect the community there. Yeah, Arshad.
Yeah, so I was interested with your aerial herbicide experiments with the BLM service. How is it kind of an issue that the herbicide may not be controlled for properly, or the amounts of herbicide in various areas where you get soil carbon, or is it controlled for? I would imagine that.
So it's always an issue when you're applying herbicide at these really large scales. So they use a pretty targeted herbicide, which should just be impacting the creosote bushes there. And so there is a potential that there's some residual in the soil. One of the things that Katie's working with is she calls them a historical chronosequence of plots, where we have ones that the fertilizer or the herbicide was applied 30 years ago versus just 10 years ago. So we can start looking at some of that longevity in the soil. We also have to consider what's in the herbicide itself. When we're measuring biogeochemistry and you've applied a chemical, you need to consider that as well. But it is supposed to be pretty targeted. Yeah, Alicia.
Jenny, after 5 years, 10 years, 30% of the the grasslands still survived and are resilient. Anything we learned why they survive in comparison to other 70%?
So there's a few different things that they've shown. So one of the things that, because this is a meta-analysis, a lot of the experiments were done differently. Some of them imposed just one global change, so just fertilization or just temperature. Some imposed multiple. And so experiments that only imposed one global change factor were more likely to be resilient than the ones that had imposed multiple at the same time. There were a lot of patterns we looked for which we didn't find, like whether the higher diversity communities were more resilient, and there wasn't a pattern there. It really just seemed, the major results were if you put more different factors at once, which is what happens in nature, it's less likely to be resilient. And it was the length of time that you've been applying these treatments. But from what we've, geographical distribution, none of this seems to be playing a role. Yeah. Yeah, Eli.
So I'm just curious, is there a way to kind of do any kind of rough comparison to your results with perhaps what the environment was like in the Pleistocene before the megafauna was wiped out?
So for like the herbivore experiments, or just in general?
Just in general, I know people have talked about how elephantids, were really important in, the nitrogen cycle, for example, in the past. So I know this is a bit of a stretch.
Yeah, I'm going to stretch a little bit here. Is it some of the things that are coming to mind? And this is in the grasslands that I work in northern Canada, one of the things that I guess vegetation or plant taxonomists have seen there is that those grasslands that grow in the glacial deposits are really similar to the Russian steppe grasslands that they think were the grasslands that were present when you have a lot of large herbivores that happen like in the Pleistocene. And so because these grasslands probably evolved with large herbivores, it's been the reason that they have started bringing back bison into these ecosystems. So trying to introduce large herbivores that they think would have been present there in the past. So I had an undergrad 2 years ago who sampled one of the First Nations communities, has some large bison exclosure fences and these types of grasslands. We've kept them out and we've been following those. And so in terms of like what these chemical cycles were at that point in time, doing that type of direct comparison, and so I have no idea right now, but I could read if you wanted me to learn. But I do think that there is a lot of remnant grasslands still that we think resemble those. And so doing herbivore experiments on those grasslands could start to answer some of those questions. Yeah, Flor.
Working with such like too much people at the same thing in long-term experiments, how do you agree with all of them which method you want to use for the long term, and actually what are the problems that you have with these people maybe, or the methodology, or just follow up previous methodology like what just 10 years ago or something like that?
So that's actually super challenging. So she asked, when I'm working with these large research groups, how we come to an agreement in terms of like what our methods are, and I think even because of the way science works, who's the author on publications, all those things are something that you need to talk about beforehand. So I talk with my plant ecology class a lot about this when we talk about different types of science, that the worm network, we got together, a core group of us, 4 experiments started the year before the rest, and we got together and we planned out how are we going to measure things, what type of treatments are we going to do, what equipment will we do, and we also have authorship agreements that we've talked in advance about, there's core data and you're allowed to sample other things that can be your own as long as they don't affect the core data. All the PIs have authorship on core data papers, but we also, we stay in a lot of communication. So we've had meetings together as a network to say who's working on what now, who needs what data, let's start sending things around. And so I've had undergrads collect complementary data for experiments that an Australian collaborator is doing to try and increase the power of the experiment. But it requires a lot of communication and a lot of coordination before you start. I think there's a similar experiment that started probably about 15, 20 years ago now called BioDepth. Similar just in that it was this latitudinal experiment where they did a biodiversity experiment across Europe. And Andy Hector was one of the leads on that experiment. And as a PhD student, I was talking to him about it because it was designed so we could get around this problem that everyone does their experiments differently. And he said it was so hard to do because it's really hard to publish something that someone else is doing the exact same thing. So everyone set up their experiment and they said, I'm going to do what you told me to do, but but I'm going to change this one small thing so mine is different and I can publish it. So then they had 9 slightly different experiments across Europe, and it made it a lot more challenging in the analysis at the end. So basically, we all signed on. We all agreed for what we're going to do. Yeah.
So I'm from a microbiology standpoint, and I wanted to understand like what you guys are doing and how do you check for how phosphorus is affecting the microbes, and have you considered doing sequencing to see if it's changing the species that's happening instead, or I don't know.
Yeah, so we actually did just hire someone who does microbial community structure, and she will be arriving in January. And so I do have ideas and some different experiments on how to bring her in so we can look at shifts in community structure. That isn't something that we've done yet. Really what we're doing now, and why I haven't done that is what I'm more interested in right now is how the functioning of the microbial community is changing rather than the structure. And it may be changing because the types of microbes there are present, but more important for me are how do they affect nutrient cycling and carbon cycling. And so to measure that, we do things like just measuring the abundance and what their nutrient ratios are. So we'll do a fumigation of the soil and measure nutrients before and after we've basically exploded all the microbes. And so the difference between those is what was inside the microbes. We do a lot of enzyme analysis, and so they're extracellular enzymes that these microbes produce. They release them into the soil, and those enzymes will start breaking down complex material to get access to the nutrients there. And so we can measure the activity of these enzymes in my lab and look at, you know, as you change plant community composition, if the enzyme activity is changing, it tells us really like the plants or the microbes, it takes a lot of energy for them to produce these large proteins. And so if they're making the effort to produce them, that means that they need whatever that enzyme is getting for them. So I'm really just looking at function, but super excited that Kelly Ramirez is coming and we can start looking at some of those other questions. Yeah.
Have you seen maybe, I don't know, maybe doing this in isolated parts, like maybe in the Pacific Northwest, little islands, like per se Orgazite, I know there's a lot of permaculture in there. Do you think changes in phosphorus or nitrogen level will change in the growth between the desert and the Alaska compared to maybe other isolated regions?
So I guess one thing I want to separate out is the isolated part of it because, well, just because some of these are remote, I think a lot of us in the department who work both in Arctic and desert environments, we like referring to these as extreme environments because we're really working in areas that there's a lot of limitations for the growth of the organisms that live there. And so every different type of ecosystem here is going to have their own factors that are limiting plant community structure, the organisms that live there, and that's changing with global change. And so I think that's why we use these globally distributed experiments, because there are going to be a lot of individual differences between all these different ecosystems, and so we need to that's why we're doing experiments consistent across all of them, so we can start looking for if there are patterns or if everyone is just acting uniquely.
So that's all. Thank you,
Okay.
Jenny, one more time. Graduate students, if you haven't signed in the sign-in sheet for the seminar, please do so. It's